Hi guys, in this video we are going to talk about cell cycle and cell division. So let's begin. What is the definition of the cell cycle? A cell cycle simply means what? This is the life period of a cell in which a cell synthesizes its DNA, they grow and they simply divide into two daughter cells. So that's what the cell cycle is. And if I ask you why do we need, why there is a need, for a cell to go for the division, the answer is given by the Hertwig, and in his Kern-Plasm theory, they simply says that Kern-Plasmic index, according to the Kern-Plasmic index, if there is a change in the volume of the nucleus by the volume of the cytoplasm, then the cell simply go for the cell division. All right. And when we talk about the cell division, we got three kind of a division: the mitosis, meiosis, and then the amitosis. Okay, now let's talk about the cell phases. Cell phases, when we talk about the cell phase, we got two kind of a cell phase. The first one is the interphase, and then the second one is your M phase. Interphase is further divided into G0, G1, S, and then the G2 phase. G0 is the quiescent stage or the gap 0 phase or the resting stage. G1 is the gap 1 phase. S is the synthetic phase or synthesis phase. G2 is the again gap 2 phase. So here in the M phase, when we talk about the M phase can be further divided into two categories. The first category is your karyokinesis, where the nucleus division is going to take place, and then the second kind of process that is going to happen in the M phase is the cytokinesis. So first the nucleus is going to divide and then the cytokinesis is going to divide. All right, so cell phase is basically what? Two, that is the interphase and the M phase. Interphase is the longest one, right, having the G1s and the G2, and M phase is basically what? Simply the nucleus and the cytoplasmic division. Okay, so all right, before proceeding further, we need to know something very basic, that is the chromatin, chromatid, and the chromosome. Okay, so what is chromatin? Simply a thin thread-like structure of a DNA is what? Your chromatin. So this is what? Chromatin, a thin thread-like structure. All right, and this is your centromere. When there is a condensation, condensation, compactness, then this chromatin is going to what? Form this. So this is what? A chromosome. Chromosome. And when this chromosome is basically after the S phase, where the DNA duplication is being done, so this chromosome is going to what? Look like something like this, where let me, yeah, so this is the what? The chromosome. So here if you'll see, initially the DNA was this, but after the S phase the DNA is being what? Copied, and the green was the initial one and then the red one is the copied one. So what is this? This is again a chromosome, but in this chromosome what you see, this is what? This green part is what? Your chromatid, and then the red part is chromatid. So in one chromosome two chromatids are there. In one in this, what you see, two chromatids are there, and at the center, what you see? Centromere. Centromere. So here at the center, this is what? Centromere. So centromere divides the chromosome. So divide the chromosome into P arm and then the Q arm. All right, so here this is the centromere. Now the thing is, so in one chromosome, as this one I'm talking about, in this chromosome what you see? You see the two chromatids, right? So in this chromosome you see the two chromatid and what? One centromere. All right, so in this image what you see, this is at the center, you have the centromere. Centromere divide the chromosome into what? P and Q arm. And just left and right of this, you'll see a complex of protein which is known as what? Kinetochore. So these are the what? Complex of protein. So in one chromosome at the two side, you'll see the kinetochore is there. So kinetochore is what? Complex of protein, and it provides the platform for the attachment of the spindle fiber. So spindle fibers is going to attach in these kinetochore, and it is going to separate out those chromatids. Okay. In this image, what you see initially, the DNA is what? Thin thread like a structure, that is the chromatin. And during the condensation process, what is the condensation process? Simply, if I just show you, this is the DNA, right? And we have the histone octamer. Multiple octamers are there. Histone octamer made up of the histone protein, and to this DNA got what? Wrapped up. DNA got wrapped up. So this is what? This portion, histone protein. This is the histone octamer made up of the histone proteins, and they are collectively known as what? Nucleosome. So initially thin thread like structure, and after the condensation they form the nucleosome, then the solenoid model, and then ultimately you get something very complex, that is the highly condensed form, that is your chromosome. All right,